Hello and welcome to the first year of our competition Microbiome Explorer Challenge. So at first I will just tell you something briefly why we created this, and then we will go for the pitching competition of selected 4 startups. First 2 will be online, and then another 2 will be on site. So why microbiome? As we can see, that basically science around microbiome is exploding a lot. So it is the second fastest growing life science field in the world. So it is very exciting. There is a lot of opportunities, not only from academic perspective of the research, but also from the commercial perspective. So we thought ourselves it would be nice to see what kind of startups are in the Europe in the early stage, what are they doing, and maybe find someone to invest. And that's why we created this kind of competition. And now the clicker is not working. Yeah, so focus areas, basically, if we talk about the microbiome, so it is, it's fermentation, it's biotics, also it's different analytical tools, such as microbiomics, metabolomics, or artificial intelligence, which is built upon them, or systemic health. If we go for the different axes, as we know, different microbiomes can influence basically every system which is in our body. So even, let's say, if we talk about gut microbiome, it can influence our skin, our liver, our joints, basically everything. And different microbiomes can do the very similar things. So the focus areas are still quite broad, and the market in this space is growing like a lot. So we know that each year it is about the 10% of the growth. So there is a lot of opportunities from the technological perspective, from the business perspective. So there can be a lot of startups that can be of interest. So we will have some of them here. Some of them use artificial intelligence. Some of them want to develop new tests for different different aspects of life. So I'm really looking forward for all of them. And so we were thinking that what can we offer? Because we can offer money, but everyone can offer money. But what we can offer as a people who created this competition is mainly the know-how. We have a worldwide network of basically the best scientists. We understand, we consult regularly, basically for whole world, even academic research, industry research. So we can offer all of this know-how to the company that will win this, which I think is even way more worthy than money itself. So this is everything that we offer. And it is basically mentioned here with the awards. And what we offer also is that we are still co-organizer of the biggest scientific conference in the world, which is focused on the gut microbiome and biotics. So the winner we can come with us, and then we can get a beer in the Krakow next year. And something about us, who will provide then let's say the counseling or how to say, or support for the winner. So it's me, and very briefly, I am just an entrepreneur in this field, so I know how to build a company from the scratch. And that's about it. And then we have far 2 more interesting people than me. We have Dr. Peter Kurti, or as I call him, Mr. Peter, who basically was leading one of the biggest companies in the world in this field for a long time. And now he's basically, what I can say, you are consultant from for Europe, Japan, and India in the biotic field. All this free. So so we can provide knowledge basically through the worldwide market. And then we have one of the top scientists in our field. You have it doesn't work again. He's there. Yeah. We have Benjamin. Jensen from Copenhagen, from Denmark, who is excellent scientist, very well established, but he's not just the academic scientist, he's active in multiple startups, so he knows also how to do very good research in a microbiome field, but also actually how to build a company out of it. Because one thing is to, to do some research, very different thing is to be successful in the entrepreneurship world. And he brings this knowledge, which is together, which is very nice. So we can really, for the winner of the competition, we can provide all of our know-how to support the startup. So we three, together with my colleague Norbert, we decided that it would be a good idea to establish new kind of competition for these early stage companies and to support some sort of microbiome ecosystem on the European level. And so this is our first year. It's still small. We just had 16 startups that actually registered. We selected these 4, and we will see what will be also for the next year. So I'm now looking forward for the pitching itself, and we can start with the first company, DataBiomics.
Okay. Yeah. First of all, thank you very much for having me. Accepted and selected me for the panel. My apologies for not being there in person. So my name is Marco Meola. I'm the CEO and co-founder of DataBiomics, a Zurich-based tech bio company. And what we want to do is we want to unlock the power of the human gut bacteria, the microbiome, as a diagnostic biomarker for nutrition and health. So if you look at the many different diseases that have been correlated with the human gut microbiome, colorectal cancer, for instance, affects 1.9 million patients every year. And incidence in the past 10 years has doubled in the patients below the age of 50. So there is an urgent need also to find biomarkers for early detection of colorectal cancer, which has been shown to be possible. Beyond colorectal cancer, of course, microbiome is connected to obesity and inflammatory bowel disease like ulcerative colitis and Crohn's disease. But it's not only linked to diseases, actually, and there are many health conditions like IBS that affects almost 10% of the human population. Nevertheless, the human gut microbiome is not used as a routine care in the clinics yet. So why is that? Well, it turns that getting actionable insights from a stool sample is much harder than it looks. It is a long process. Usually, it takes up to 3 to 4 weeks. For practitioners, it has a high workflow friction. It is also very expensive for the patients. And last but not least, it's very hard to get actionable insights from the results, which are very complex to understand. So that's where DataBiomics wants to come into place. What we want to offer is a end-to-end platform for nutritionists with a very low workflow friction, where basically they can order stool kits through our web app, which we will be shipping to their customers. The kits will be shipped to our partner lab that will be sequenced, and we will receive the raw sequencing data that go through our validated automated platform, where we can leverage the results back to the web app that the nutritionists have access to. We want to integrate RAG-based AI assistants to actually combine the whole global knowledge of the microbiome related to the results of the patients, and then generate automatic reports and, of course, a coach, an AI-based coach, where the nutritionists can ask further questions and eventually generate reports. We want to be 50% faster than it is today. We will be reducing also the workflow friction, having this web app and having highest resolution of results through our web app and species resolution that we can achieve through our web platform. Oops, wrong direction. Okay. How we want to get to the market. So we want to address nutritionists directly through our web app, but we also want to give access to consumers to order actually the stool kits. But by ordering the stool kits directly, they will have to select the nutritionist that will get the result. So we will be not offering the results directly to the end consumer. We will reach out to early adopters as nutritionists who understand our kit, and so they are more able and prone to also offer it as a diagnostic kit. And the web app will be on subscription base. So that means that the nutritionist basically pays a subscription and then can order the kits. The higher the subscription they pay, the lower the price kit for the nutritionist, the higher the revenue for the nutritionists themselves, which they can resell at their own retail price. As already mentioned, the market is growing fast in the microbiome space, both in nutrition and health, with a double-digit CAGR. And the market we want to tackle first is, of course, the one where we are in Switzerland. But if we look at the European market of nutritionists, then we realize there is an increase of nutritionists by 6% every year. So there is increasing awareness about health and nutrition, and people actually are higher educated, so they really want to have insights into their health. So the business model for nutritionists, as I mentioned, is going through the web app, which is in development. But we already can offer our bioinformatics or our pipelines as a service to small, medium enterprises, CROs, and diagnostic labs, where we basically can do biomarker discovery. This is something that we already operative on and how we get traction for. Regarding the research testing markets, there we reach our customers through direct sales, marketplaces, and partnerships. And these are mostly a B2B market business model. So at the moment, we already have traction. So it means we had already executed contracts for 2 customers. We have some more potential customers in the discussion, and we have been reached out even from a US company for an even much bigger volume of service that they asked us for. For the nutritionist market, we plan to develop the web app in 2026 and having a first version by early 2027. So, of course, at the beginning, the revenue will be much lower, but we hope to get a fast-growing market revenue over the years. What worth mentioning here is that we estimate a gross margin of 60% on our service. There, of course, there is a competition around as well. Of course, what we think we can excel here is that we do not offer direct-to-consumer kits, and we also always go through the nutritionists. So we always have a human in the loop. We offer a subscription-based web app, which is something quite unique, where the other competitors rather offer PDFs as a report. We can achieve species resolution even with targeted sequencing. Those who achieve species resolution usually use shotgun, which is more expensive and has also the downsides of sequencing the human genome. We have a patent filed, and yeah, we want to also include literature-based AI assistant, as I mentioned before. So the company was incorporated this year as soon as we had paying customers. And what we plan to do is now fundraise for a pre-seed by the end of this year, and then also developing the web app in 2026 to then go live with the web app end of 2026, early 2027. The team is composed by myself. I have 15 plus years experience in the microbiome space, in clinical, environmental, and food microbiomics in both academic and accredited hospital settings. My co-founder, Dr. Matthias Dreyer, is a bioinformatician with 5 plus years of experience. In the meantime, he has turned into a software developer. And we are supported by Michel Lehmann as a fractional bioinformatician. And then we have awesome advisors from the Fachhochschule Zürich, from ZAW, Dr. Adisa Trinjanin, and Ivana Vukovic from UABS. And we are also supported by Amichai Tömim as a strategic capital advisor. Yeah, that's it for the pitch. Thank you very much for listening and then opening to your question. Thank you.
Can you hear me?
Yes.
Yes, you can. Great. Thank you very much for the presentation. And I have the first question. You mentioned patent several times. Can you elaborate on what exactly is patent for, or what do you want to patent?
So we have filed a patent to actually be able to have a higher resolution in the identification of the bacteria, so that we can achieve strain resolution.
Okay, thank you very much.
Yes, sure. Very nice presentation. This is the presentation level we wanted to see. Okay, just one question. You said 50% faster than others. What does that mean in days, hours, weeks?
Yeah, so the thing is that very often nutritionists in the whole workflow, there are samples that they ship that get stuck at the border, and there are many other, let's say, hurdles. So that as of now, for most of the samples, it takes 3 to 4 weeks. So we want to really get it to 10 days, maximum 2 weeks. Of course, there are lab-based limitations. So in order to get it faster, it can be done also within days. But then, yeah, the cost could be higher.
Okay, 10 to 14 days.
Reduce it to 10 days.
10 to 14 days, you said?
Yeah.
Okay, then don't you limit the indications where this data can be used? I mean, who stays longer than 12 days in a hospital? You assume it is for hospital patients, right?
No, we offer it to nutritionists.
Okay, you go to the nutritionist. And who are the clients of the nutritionist? People who sit home or people who are in the hospital?
Usually people that have chronic diseases that are not, or chronic conditions that are not treated as diseases, that maybe went to the doctor but it didn't help, then they go to nutritionists, try to actually solve their problem with nutritionists. They are not holding a hospital. We don't go into clinics quite yet because of its accreditation and regulatory assets, which makes it more difficult to get in at first.
Okay, I'm concerned about the microbiological qualifications of a common nutritionist. So what is the actionable conclusion out of the data? I mean, most nutritionists have no idea about microbiology. There are some enthusiasts, of course, who are really top-notch, but most of them cannot really conclude anything out of your even perfect data. So how do you deal with that?
Yeah, that's absolutely right. In fact, that's why we actually provide the microbiome profile of the patient, but that's not enough for the nutritionist. And that's why we need AI to basically translate the microbiome fingerprint of the patients into something like dietary recommendations that we then leverage through AI to the nutritionists.
Okay, so we arrived at AI again. What do nutritionists think about AI or medical profession? I mean, do you really think they would follow AI's recommendation? Not in the future, but nowadays, let's say next 3 years.
Yes. So nutritionists, they want to see the proof that it's science-based, and we will have to generate trust that what we actually have developed is kind of certified to a level that they can trust. But I mean, this is definitely a thing to consider, is how much they can trust it. But I mean, there are nutritionists who are more open to adopt these new tools, also because it extremely lowers their workflow friction and it generates more insight. And that's the nutritionists we want to target at first.
Okay, thank you. Benjamin, you have a question?
Yeah, thank you for a very nice presentation. I have a number of questions, but so I would have really liked to see your interface, but that's under development. So that is something that we don't know yet. But can you allude a bit do you have any experience in developing that, and how do you foresee the interface will be built so that you will differentiate yourself from the current competitors in the field?
Yeah. So the web app is in development, and it basically is a way to dynamically visualize results from the microbiome, which as of now are basically leveraged as PDFs. And what nutritionists have told us that they often have hard times comparing samples from the same patients over time because they basically get always a PDF. This is something not really deep tech in this sense. So already having a web app where all the information of the patients and their microbiome is stored is an asset. And then the other thing is connecting this to AI. So the AI is something that we are developing with the Fachhochschule Zurich. It's a RAG-based, knowledge graph-based LLM on top AI system where we basically have structured and unstructured data from the literature. Where we basically extract the relationships, and then we try to connect it with the microbiome profile that we get from the patients to basically then generate through the AI. So there will be many different AIs within this system. The report to the nutritionists. So that's the vision. We will be starting with the web app, maybe first without AI, because this is something that we really have to develop first. So
Okay.
it's not.
Thank you. Yeah, there is another company. I'll just want to bring it to your attention so that you can potentially either get inspired or see how they have done it. They are called Unseen Bio, and they have sort of the same mission. They are based in Copenhagen, where people can interact with their own microbiome in an app. Highly intuitive, and then they can pay additional fee, and then they can have some 3D goggles and so on, so they can basically move around between their own microbes. So there are some quite fancy tools, and there are a number of companies coming up with these ambitions. So I think it's important that you sort of become very aware of what the competitive field is, so that you either have an edge or just have a different location. That could also be, but just so that you have a clear strategy on how you will relate to those things. But then talking about AI, so I want to jump on that first, and then I have something about the resolution by the end. So AI, what is it trained on? So my concern would be that AI is only as good as you train it. And if you want to train it on the literature that is published, I would argue that the far majority of what is published is noise. Because there are so many publications coming out, and there are so many things where you have seen something going up in one indication, small sample size, whatever, then depending on what journal and so on, then you can publish that. And if the AI trains on that, how will the AI be able to see what is your microbiome, what is mine, how can that be sort of shifted toward a more beneficial phenotype?
Yes. So to the question of the literature, that's true. So there will be some kind of a weighting system that basically tries to, correlations that have been seen over and over in many different publications will have a stronger weight than others. Regarding the comparison of the microbiome between people, I mean, what it is about, it is actually to understand which metabolic pathways are covered and which are maybe underrepresented in the patient. It's less to compare the patient with other patients, but it's on a metabolic pathway system that we want to go to. And to build this, we actually have to build all the ontologies that links the microbiome ID to metabolic and functional capacities. And this will be done through a knowledge graph that actually connects all these ontologies and entities within the system, from the ID of the bacteria to actually the metabolic capacity and the pathways.
Okay, very good. And then in terms of you have the subscription strategy. What would motivate me as a customer to subscribe? Normally, when you subscribe, I would assume that I have it on a repeated basis. So would that include that I would need to have stool kits from you and then sample over time, or how do you foresee that? I didn't quite grab from you.
Yeah. So the subscription is for nutritionists. So nutritionists can, but they don't have to subscribe. If they do subscribe, every time they order a kit, the kit has a lower price than the full price.
Really? So
They can resell it at their... Sorry?
It's not for the end user. It's for the nutritionist that is in the middle.
Yes.
Okay, then it makes
And then can resell it at their own retail price, and the delta is their own income.
And so if I have time for two very short questions. One thing is the resolution. You talked about species, but you also talked about strains. Is it 16S long read, or how do you said it was not shotgun sequencing? So how do you come to that resolution?
Yeah. So regarding the state of the art now, we are at species resolution with short read sequencing. But of course, with long read, you would achieve species resolution with no problem. It's just more expensive and not really to be used in practice. So we still are on short read here. For strain level resolution, it just allows to narrow down the metabolic capacities as well of the different strains within the same species. How this is done, this is still not disclosed. It will be then disclosed once the patent is published.
Okay. And in terms of the stability of the stool sample, when you collect that, what considerations have you done? And then lastly, you can just answer at the same time. And then lastly, what would the cost be? Because you said you would be 50% of the average competitors, if I remember correctly.
Yeah, for the turnaround time.
Yeah. But what about the cost would also be cheaper, right? Or did I misunderstood that?
So this is something we're still in discussion, because there might be 3 different services. You know, you can actually offer even 16S and shotgun, and then at different prices if this is requested. The price we try to be around 250, maximum 300 euros. As a full price, as I said, but then actually it heavily depends on the this is how the nutritionists will then sell the kit. It depends on them.
And the stability of the stool sample when
yeah. Well, this depends on the kit that we will be using. We haven't defined that yet.
You haven't defined that yet. Okay.
Okay,
Thanks.
guys. Time is up. Actually, I had a couple of questions, but Benjamin, you were nice enough that you asked them instead of me. So I guess it's over. Thank you very much for your presentation and answering all of the questions. It was very interesting. And now, as time is up, so we need to go for another startup, Trillion. So can you put their presentation?
Mm-hmm. What will do? Can you see it?
Yeah, we can see it. Looks great.
All right.
So, floor is yours.
Thank you very much. Good morning. We are Trillion, and we are delighted to be here today with you. And we will show you how we are rewriting human resilience from within. We have built a platform to discover and develop precision bioactives that reprogram the gut microbiome. Our mission is to restore brain and immune health by targeting the root causes of unhealthy aging. We believe, and we will show you today, that we are next step in microbiome precision modulation. We are all facing a hidden crisis. Our modern lifestyles, processed diets, chronic stress, our environment are systematically destroying the gut ecosystem that our health depends on. This gut ecosystem collapse leads directly to chronic low-grade inflammation or inflammaging. This inflammaging is now understood to be a core driver of the major chronic diseases in our time, from cognitive decline, depression, to metabolic disorders, and the rise in autoimmune diseases. We are all busy treating the symptoms, but we are failing to address the root cause, the collapse and destruction of our inner ecosystem. Our solution is to rebuild that ecosystem from inside. We do this through the selective induction of keystone species and the critical metabolite that they are producing. To be clear about what we are a platform that identifies precision molecules that reprogram a person's own native gut microbes. This is all validated through advanced human gut models and our proprietary machine learning pipeline. This gives us a modular, scalable platform to address cognition, immunity, and longevity. And it's also just important to clear what we are not. We are not generic fiber or prebiotic blends that act as blunt force fertilizer. We are not probiotic supplements, which can be fragile, transient, and often struggle to colonize and engraft. And we are not generic botanicals and vague claims. We are moving beyond the current generation of tools. We are not fibers, we are not probiotics. We are precision molecules that reprogram your gut. This is the engine that makes our discovery possible. We call it our symbiotic discovery engine. It starts with a unique and sustainable blue ocean source, upcycled plant waste streams, which are rich in complex unexplored bioactives and also provide a very low cost of goods, which is important in consumer space. We then identify the most promising compounds using our proprietary industry-standard human donor models, human gut-not-shape models, and AI-driven analysis. This is where we see the unique mechanism of action in the gut. Once the lead compound is identified, we additionally validate it in robust clinical data packages for specific application, like gut health, cognitive health, mood modulation. Finally, we develop a product, this validated patent-protected bioactives for multiple high-value markets, as for example B2B ingredients, for supplements, or with more clinical data also as medical foods. This isn't a single product. It's a scalable, repeatable engine for creating a new class of health assets built on a foundation of sustainability. So how does it work, and does it work? The answer is yes. This is the data of our patent-filed lead asset, TR01, targeting the gut-brain axis. We are the first and only one to demonstrate such a strong targeted induction of these critical keystone species. So these are obligate anaerobes, which make it very, very challenging to develop probiotics from the bacteria, because if you take them out of the gut, they die very quickly. We see a 45-fold increase in Agathobaculum butyrici-producents, which is known to be strongly neuroprotective and strongly negatively correlated with developing Alzheimer. We see a 15-fold increase in Copococcus catus, a critical species linked to mood and well-being, as it produces vitamin B1 to B12, dopamine, and propionate. And we see a 9-fold increase in Anaerostipes harderus, which is essential for strengthening the gut barrier. And interestingly, it also transforms harmful D-lactates, which you get from unhealthy diets, too many carbs, into a highly beneficial butyrate. So these are not random bacteria. They are targeted functional keystone species essential for controlling neuroinflammation and improving cognitive health. This is what precision looks like. We also increase, here see on the upper right, short-chain fatty acid production, butyrate, propion, acetate, 150%. They are linked to cognitive function and less inflammation. And we can also, as a byproduct, boost resident probiotics like bifidobacteria twofold. Our science unlocks a massive opportunity. We are targeting the 24 billion B2B gut, brain, and mood ingredients market, which sits within the much larger 480 billion nutrition industry. Our go-to-market strategy is capital efficient. We start with B2B ingredient sales to partners like the one you see here on the left, who are actively seeking validated next-generation ingredients. And we have now 5 letters of support to combine our ingredients with their products. This de-risks our path and builds revenue. From there, we expand to white label products and ultimately strategic licensing and potentially even therapeutic application to a spin-off. Based on our B2B sales pipeline, we project $75 million in occurring annual revenue by year 6 and building a company with a value of about $350 million. Why is this also critical now from another perspective? Because in the supplement industry, there's a credibility gap. Up to 80% of ads lack scientific authorization. Many products simply don't work. Consumers are skeptical and B2B partners are desperate and really require a real differentiation and science-backed value. Trillium is the differentiation. Our science provides precision, human-validated data. This builds trust, giving our partner the proof they need for a transparent label. And this creates impact, allowing them to launch premium, category-defining products that cut through the market noise. We are not just selling ingredients, we are selling scientific credibility. When you look at the competitive landscape, it's clear what we aim to win. The market is full of generic, non-selective fibers and probiotic-to-bake claims. Even advanced prebiotics are just food for the microbiome. Trillium is a new category. We are precision targeted with specific evidence mechanisms of action. Our advantages are clear. We are beyond fibers using bioactive small molecules. We have proven gut-brain impact. We activate the native flora in your gut, and we have a sustainable upcycled source. And we are protected by IP. This is our blue ocean strategy. And we have a strong IP strategy as shown here. We filed our foundational patent in May, protecting our lead composition, compound, usage claims, and our manufacturing methods. This is now being expanded to cover novel symbiotic pairings of our ingredients plus existing ingredients, and critically also developing our AI engine further, which will probably remain a trade secret. None of this possible without the right team. My co-founder, Tim, who was here at the beginning, and I are joined by a world-class team of scientists and advisors. Tim and I combine over 30 years of experience pioneering microbiome science and biotech commercialization across pharma, small biotech, large biotech, and startup roles, with deep industry experience from leading institution companies like J&J, DSM, Bayer, and H&H. We have the scientific and commercial experience to execute this vision. And we have significant momentum. We have backing from key Swiss and EU innovation agencies like InnoSwiss and EIT, and we've been recognized with 3 major awards this year alone. We got a Probiota Pioneer Prize for Americas for new innovative products. We got the 2nd prize in the ARDD, that's the biggest aging research drug discovery conference in the world for startup. And we also got the 1st prize in EIT Food as a Medicine for our product we're developing. And most importantly for our business, we have strong industry engagement with 5 letters of intent already signed from major strategic partners. This brings us to our plan. The financing here is probably not so important in this context, but this shows you also a bit the regulatory approach and go-to-market. Right now we are raising a total of 3.1 million seed round, which can be deployed in 2 milestone-based tranches. On the left, in yellow, the initial 1.3 million bridge is entirely focused on the critical milestones you understand best, really finalizing at-scale GMP manufacturing process, completing a pivotal safety and tox study. Our extracts come from food plants, but it's still an extract, so we need to show that it's safe, and filing our self-affirmed GRAS to unlock the US market. This first tranche gets us to go-to-market readiness. The second tranche will then fuel our commercial launch, ramp up B2B sales, and fund our first in-human clinical trial for gut health. This is capital efficient plan to get us to the first revenue and full regulatory validation. And the second step will, of course, then go to filed FDA GRAS. And with Series A, we also want to go for novel foods in the EU, but we know this will take more time. Well, this is the last slide here, and Trillium is at the intersection of a massive market and breakthrough human-validated science. We have a world-class team, a scalable B2B path forward, and a clear de-risk plan to get there. We are now raising our seed round to scale production, complete our safety validation, and launch. We're the next step in microbiome modulation, and we invite you to join us in rewriting human resilience. Thank you very much.
Okay, thank you very much for a very nice presentation. And I will start with the questions. You mentioned several times that you do some sort of precision molecules. Can you name some of them? Are there, let's say, metabolites from the bacteria, or the polyphenols, or what kind of molecules should we imagine that you want to produce?
The plant extracts for qualifying, yeah, they're small molecule, polyphenol, flavonoid, tannin-like small molecules related to this class of molecules. That's what we focus on, because they're the most interesting novel activities and the most powerful impact really on the gut microbiome.
Yeah, actually, I agree with that. Also in our experience. Next question is that you mentioned you have patent. You went through it briefly. So this patent you already achieved, or is it just signed? And can you elaborate more? What is it? This patent covering?
Yeah, it's filed. It's not granted. This will take a little bit more time. So it protects key composition. It's our new ingredients, but then also combination our ingredients with other ingredients, a symbiotic activity. Usage claim, both therapeutic as well as in the consumer space for cognitive health. And also manufacturing methods. How do you get these active compounds out of the plant waste streams?
Okay, thank you. And there was, I think there was mention just how you validate this kind of, let's say, you take polyphenols. How do you do the validation? What do you do in vitro? What do you do in vivo?
Yeah, okay. So we don't really start with qualified polyphenols. I mean, we can compare this with polyphenols in the market, of course, because most molecules are related to polyphenols, but not just. We really make an initial discovery. So we take a plant waste stream and fractionate this down into subfraction and even into pure molecules, and can then test these fractional pure molecules. First, we go in vitro stool donor models. So these are stool samples from different human, old, young, male, female. You can incubate this in vitro and see what's the impact of your ingredient candidates on this microbiome. And this model has been published to be highly translational later on to clinical study of what happens in the full human being.
Okay, that's clear. Thank you.
Peter? Okay. Nice presentation. Yeah, one question. What kind of side streams you use for the isolation of these molecules? Is it available in your country, or are you importing it from the end of the universe?
At the moment, it's the end of the universe because it makes a very nice marketing story in the US and Europe. So we started focusing on side streams in the APAC region, so exotic fruit plants. What is, of course, necessary, we only go for side streams which are available in really large amounts because we want to have a scalable manufacturing process. We have now a supply chain in Thailand, the CDMO in Thailand, that is able to manufacture that. And it makes a nice story then in the EU or in the US. Some of these plants are not well known. It's a nice spin around exoticness. But we can do this the other way around. So, for example, we are looking now also at fruit peels from Swiss waste streams, apple, pears, other fruits which are being thrown away. And then you can actually go to also into the APAC region, and it's exotic for them. So Swiss alpine extracts in the APAC region. So exoticness can go in both directions. Important is that these waste streams are present in really large amount that we can scale up.
Okay, thank you. You said you ask 3.1 million euro. What do you offer for that?
You mean equity-wise? Or?
Yes.
Okay. So the first tranche has a lower cap, 6 million. And the second tranche will have a higher cap, 10 to 12 million, because then we already went to the regulatory approval and the pivotal safety studies. So at the moment, we're aiming for convertible notes. So if you're asking what we're offering, these are kind of the cap we're envisioning.
Okay, no problem. I expected that reply. Thank you. Okay, we will have a look at it. Okay. Benjamin?
Yes, thank you for a very nice and clear presentation. You might have said it and then I missed it, but when did you launch or when did you establish the company?
The company was incorporated in March 2023, and we started really with bootstrapping. So the company is fully bootstrapped. That's why I also now need to do the fundraise. Tim and me put in our own money, and about 1.5 years ago, we started with that work, paid CROs to perform the test, had the collaboration with the University of Basel, the Department of Pharmacology. So it's fully bootstrapped. We own all the equity at the moment still ourselves. So 1.5 to 2 years the company has been running now.
All right, thanks. And the data that you showed, where you have an increase of select bacteria that you showed us, was that in human subjects? Was it human microbiota in vitro? Or was it animals? Or where did the data?
It's a question. It's because we say it's human data, and it is human data, but it's human stool samples. So pretty much you take stool samples from different humans, young, old, male, female, and you can incubate this in vitro an anaerobic condition with all the nice goodies, which simulates the content of the gut, because you have partially digested stuff, fibers, dead human cells, the entire microbiome. See, this makes for an extremely translational model. And it is human data, but it is in vitro human data, which sometimes is a bit confusing.
Yeah, exactly. So, but how do you envision, and have you tested it in terms of stability? Because I assume that you would administer it. If I were to take the compound, then it would be oral administration. And a lot of things is happening in the ventricle and the small intestine and so on before it reaches the microbes the anaerobic microbes that you're targeting here in the colon. So in vitro, of course, you can just add it. You can have all the right culture medium and conditions, but then you just add your substrate. But what happens when you administer it to a live human being?
This we'll see. These are small molecules. We can test for certain key parameter stability, like we can acidify and see would it be on its own able to pass through the stomach. If you have compounds which would be really sensitive, they probably would need to be put in a pill which protects them in the first stage. But most often now, especially also the lead ingredient, seems to be extremely stable. A small molecule, almost indestructible. And probably it will be, especially with partners, really combined with other products. Like we have a lot of interest from companies selling fiber products and probiotics because it synergizes very well. So probably it will go, at least in the supplement side, into a pill. But still fibers are most the bulk of the pill because you need a lot of fiber to have any activity, and maybe our ingredient, maybe 50 or 100 milligram of this pill. So at the moment, everything indicates these compounds are extremely robust, but we will see when we do the first trials in full humans.
Thanks. And do you know about because you talked about which microbes that I assume that you selected the ones that you would like to highlight that increased, because a lot of things is of course happening. So do you know what is the bacterial load? Is that changed? And if not, if that is stable, what goes down? If something goes up relatively, something must go down.
Yeah. No, it's the total microbiome stays more or less stable. It's actually quite specific, or even very specific. So it's difficult to show all the data here, but what you saw, this massive boost, these are these 3 species we wanted, this network we wanted to boost. You see a mild increase of bifidobacteria, it's about 2-fold, and you have a few species which you would not like to have in the direction of E. coli, which get repressed, which kind of makes sense if you think we're modulating this gut microbiome, and a different way of doing that. So certain effects are direct. We don't think that the effect we see is a direct upregulation of the bacteria I showed you. We think it's indirect. That it's a combination of repressing competitor species and providing actually also metabolites from intermediate species that will feed the species you see there. So the story is a bit more complex, but we can rip it apart with our discovery engine. But really, it is what you saw, this massive upregulation. These are just these 2 species. And otherwise, it's on the rest of the gut microbiome, there's actually not so much happening.
But
so
you must have some repression, as you allude to, because I think if you summed it up, the 3, it was a total of 15%, almost from 0% to 15%, 4-5% of those 3, which means that must go down.
Yeah, it's a fold induction. It's not a percentage of the mass.
Okay. I thought I read it as the fold induction what you wrote, and then on the x-axis on the y-axis you had percent as far as, but I might have misread it.
Yeah. Let's hop quickly there.
Yeah, guys, this is like.
So it's a fold induction versus baseline.
The 45, 15, and 9 is fold induction, but I assume that the numbers you have on the y-axis is relative abundance, isn't it? From
that's log fold induction on the y-axis.
All right. Okay, makes sense. Good.
Okay,
thanks,
guys. The time is over, so I have to interrupt you.
Thank you all.
So thank you very much for your presentation. And we are now moving forward, and we are going for live presentations of the startups that are here. So now we will have like 3-minute break. Yeah, or can we continue? Or we can continue directly. So that would be great. So now I'm giving microphone to Optibiomics, and Floor is yours. Enjoy.
Hi, everyone. Do I have a timer somewhere so I know what's happening? Or I just start and see what's happening? Okay. So my name is Anna Melvan, and I will present to you our project, Optibiomics. So the problem today is that 75% of people with gut issues remain undiagnosed until they have a severe diagnosis. So if you come to a doctor office with bloatation, diarrhea, constipation, cramps, anxiety, skin conditions, like in 75%, you will be sent back home unless you have a severe diagnosis like Crohn's disease, cancer, or something like really serious. So this is happening because the health data and microbiome data is super scattered. The diagnostic methods are outdated and focused on severe diseases, and the microbiome tests today, they give you the narrative report instead of an action report on what you can do to feel better. So that's why we built Optibiomics. So Optibiomics, it's a digital precision nutrition tool where we collected and integrated a huge amount of health data. We developed our own proprietary algorithm. It was actually part of my PhD, the algorithm part. And we built a user-friendly interface on top of that. And as a result, we provide clinicians with 4 to 5 actionable steps on what a patient can do to feel better. So this is a product in a nutshell. We like to think of ourselves as Google Analytics for gut microbiome. So the same way that Google Analytics analyzes and tracks websites and tell you what you can do to improve your website, we do the same thing with a gut microbiome. So I'm shaking. Okay. So how did we do this? So we integrated really big. We spent like 7 years on collecting and organizing unorganized biological and health data. So we fed algorithm with diseases, medical history of a patient, food databases, microbiome databases, which microbe likes to eat which food databases. We also took into account the mental state of a patient, like we take the mental health scoring, and also take into account the drugs that a patient is taking that's blocking certain microbes and doing the dysbiosis in the gut. So we fed, do I have the thing? So we fed all of that into the algorithm and learned the behavior of bacteria. I'll show you later that. And then when the patient comes to doctor, they do the gut test. We get the raw data from the lab, and we can actually do it with any lab that provide us the reference interval of bacteria. And then we match that with the knowledge and provide a clinician with 4 actionable steps: personalized nutrition plan, supplements recommendation, drugs recommendation, and additional diagnosis needed in percentage. So how is this all happening? So I'm a mathematician in a background, and I did PhD in bioinformatics. So I did the modeling of gut bacteria, and like the biggest problem was that the gut is super dynamic and it's changing, and most of the data we have is it's a static snapshot of a moment what's happening in a gut. There were some longitudinal data, but not enough to learn from them. We tried to have our own data and to grow the microbes in vitro, but due to nutrients, like not all bacteria survived, so we kind of got the biased results. And then 3 years later, we decided that somehow we have to learn from the static data. So what I did there is there is this ecological model from 1960s. It's called Generalized Lotka-Voltaire model in the background. And basically it's this predator-prey model used in ecology, where if you take a snapshot of a jungle and count the animals in a moment, you can predict how many animals will there be in a jungle 1 year from today, based on the resources, based on the number of animals, and so on. So the only difference, small difference, 2-year difference here was that we don't know the absolute numbers of bacteria in a gut. Like we don't know what is the definition of a healthy gut. And also different to a jungle is that the animals in a jungle can always move somewhere else, whereas in bacteria, whereas in microbiome, the number of bacteria will always remain the same. So this picture here is like 5 years later. It looks really simple, but it was really hard to get to that. And here I will show just this is on a film level, so it's easier to notice the difference. So the nodes are representing bacteria and the edges are representing interactions between bacteria, meaning the blue line is positive interaction, meaning one bacteria grows, the other one grows, and the red one is negative, meaning one grows, the other one is dying. So this was the clinical trial on obese population, and we built network model on lean population and on obese population. And just by looking at a photo, you can see that there is like a significant difference between in a behavior of microbiome within these two populations. So how are we using this today in a practice? So we get the report from the microbiome lab, feed it into our software, then we match for each bacteria to its reference interval, pretty much the same like with a blood result. And then I'll just go back here. And then we provide a food to feed bacteria instead of feeding the patient. So why food? Is because once we have the network, then the question becomes a question from network modeling, which is what is the smallest change needed in a network to shift the network towards the desired look of a network, meaning what is the minimum change to maximize the change of a network. So we provide a patient with a list of foods. Then if they don't want to eat the food, then we give them supplements, or if they have 0 of certain bacteria, then we also give them supplements, first to put bacteria in a gut, and then we'll give them food. So basically it comes to 4 or 5 actionable steps that a patient can do straight away. This is a case study. This is another clinical trial we did last year on mental health. So, the assumption was that we can affect the mental health of a patient only by changing their diet. So I'm not saying that if you have some mental issues, just change the diet, like you would have to change lifestyle and psychotherapy and so on. But we didn't want them to change anything except the diet. So this is an example of a patient. It was 38-year-old male. He was healthy, not taking any drugs, but super anxious. So we did a gut test, did a food intervention for them for 6 months, and then another gut test. So this is the results. So in the beginning, his microbiome diversity was low, so he had 3.0, whereas 6 months later, he came to the healthy reference range. And for species richness, in the beginning, he had 91, and later on, he had 102, and the healthy interval for bacteria is 93 to 128 species. So he could have fixed his microbiome even more, but it would require much bigger lifestyle change and much bigger diet change. This was just like introduce these 5 things in your current lifestyle. So we checked the markers for gut-brain communication. The markers we got from GM Repo is the repository of gut bacteria clinical trials, where they gave markers for certain diseases with a probability of that marker affecting disease. So in a first measurement, 3 out of 5 bacteria were out of balance, and then 6 months later, 4 out of 5 bacteria were in balance. And again, the 5th one could go into balance also, but it would require bigger change from him. So this is the market. We believe that we are part of clinical decision support system market because we are not here to change a doctor, but to provide a knowledge to a doctor so that he or health care practitioner so that they can diagnose their patient more effectively. I will not now go into how we calculated our sum, but I can tell that later. These are the competitors. So out of the direct competitors, there are 3 companies doing B2B microbiome diagnostics. Only Enbiosis, it's based in Europe. The Microba is in Australia, and DigiBig Health, they're now in US, but they're slowly shifting towards Europe because they entered UK last year. But what differentiates us the most from them is that all of them are just checking your gut test and then give you a food plan. Whereas we take into account the food restrictions you have, the drugs you're taking, and also disease that you already have. Meaning if you have high blood pressure or heart disease or something, you already are restricted with food. So we will take like intersection of all of this and provide you with list of foods that fulfill your needs. So our business model is B2B SaaS. We are almost done with MVP. It should be done by February. We have 4 clinics we are developing this with, like a pilot partners. All of them are private clinics because we still didn't file for CE mark, so we cannot enter the public health until doing that. This is the team behind the project. So I'm a founder and CEO. I'm a mathematician having the PhD in gut microbiome. Then there are 3 more co-founders with me. Irena Šoljić, she's a... Should I stop or say this until... Irena is... PhD in food science, and then Marco and Josip, they're both mathematicians doing the computer science. Marco is lead architect and Josip is doing the cloud magic. Together with us is actually 11 team members now because we got seed round 2 months ago, so we hired 2 more developers and 1 marketing person. So that's basically it. We are connecting body, mind, and data to optimize patients' care. Thank you so much.
Does it work? Yes. Thank you very much for the presentation. And now with the funny part of the questions. So, do you focus only on the abundance of the bacteria, or do you take into consideration also like molecular pathways?
No, just the abundance.
Just the abundance. Okay. How do you actually establish the reference point? Because usually it is like every lab does make their own reference points and it's a mess. So how do we do it?
We noticed it's a huge mess. So we did statistics on a human microbiome project, American Gut, Twin Study UK. So basically we are taking the technical part, which sequencer are they using and so on. So we try to fit with that. It's not working really well. So then once we have a collaboration with a lab, we ask them to unlock the reference interval for us. Some wants to do it, some not. But the ones who don't want to unlock the data, they're willing to provide information if bacteria is low, normal, or high. And we can work just with that information so we know which direction to shift the bacteria. But like 80% of them will unlock the data for us.
Okay. You mentioned that you can also, like, let's say, suggest some sort of supplements. Do you also plan to produce these supplements, or you want to say go and buy this in pharmacy or whatever?
We have our own prebiotic, but we will not offer it's for the Butyrat bacteria. We will not offer it to you unless you need to grow your Butyrat bacteria, and the rest we are just sending you to the one that are already on the market, because we kind of want to be just like a software data company.
Okay, that's clear. And also for the diversity, which you mentioned, what do you use? It is just like a Shannon index, or how
Yeah.
to?
Yeah.
Okay. Okay, that's clear. Thank you very much.
I have no question. I have to digest. That was just very deep for me.
Benjamin?
Sorry. Yep. Yeah. It's I just had. Sorry. Interesting presentation. There was a few fallouts on my connection here, so I'm not. It could be that you mentioned it and I just missed it. So I apologize for that if I pose a question that you already answered. But how? Yes. Yeah. Can you hear me now? Is it better?
Better, yeah.
Perfect. Thank you very much. So you start out with a test, and then you use your Google Analytics software, essentially, that you have adapted for this to predict whether you should change the microbiome. I think it's a really sort of bold action that you have, and it's great to hear that you're also factoring in the medicines and nutritions, the different diets that people are actually consuming. But how do you differentiate it between different interior types? So it's if you don't have sort of the genetic landscape, you don't have the baseline healthy before the person developed any disease. If you don't have that from a subject, how would you know which direction to push the microbiota in a certain individual?
So we learned it was part of, if I heard massively, so hopefully I understood the question. So the data part was part of my PhD. So luckily they unlock a really big amount of data during my PhD because it was for the science. So from there, we learned the behavior and the reference for the healthy population. And now commercially we are just using the learning and not the data. It's kind of gray area, but it's working. And I talked with CMARC people also in Germany, and they said that it's fine. So when I wanted to learn what is the healthy population, I wanted to put all 6 big microbiome databases together. That didn't work. Then I wanted to learn a scalar between that. That also didn't work. And then we learned separately on 6 of them. And if something appeared the same in 4 out of 6 of those databases, we claim that that's the healthy thing. And like it passed, we published in Frontiers yesterday. So not yesterday, last year. Sorry.
Okay, thank you very much. Thanks.
Also, I have another question because I omitted it when I was looking through it. How do you actually do this personalization of nutrition? I don't know, let's say you measure that there is not enough Bifidobacteria or Coprococcus or whatever coccus is there. So how do you base that? What should you eat?
So our nutritionist spent 3 years, I think, on reading the science papers and then writing this microbes likes to eat this food or doesn't like to eat this food. So it's mostly on foods. We just started 6 months ago with nutrients. And then if some bacteria, I don't know the names because I'm the mathematician, but if some bacteria likes, for example, omega-3, then we will, from Frida database, it's this publicly available Danish database, we will offer the food with the most abundant of omega-3, for example. So it will always be the foods, but if you have 0 bacteria of Coprococcus, for example, then the food will not help you. So first you have to take probiotic to put the bacteria there and then combine it with a food so that it stays in your gut. I'm not sure if I understood you correctly.
Yeah, you did. And I have one business question because you mentioned that you want to go for basically B2B2C model. And for what I understood, how it results looks like, why can you do also B2C? Because people can see that you should eat whatever. You don't need a clinician to tell you to eat a broccoli. You can read it and eat a broccoli.
We started B2C, so we worked 3 years B2C. I honestly got really tired working with people, especially sick people, and then I'm not a good salesperson, and then I end up being the free psychotherapy for 2-3 hours, and then in some point I was like, no, we'll just do the software and do B2B and provide this knowledge to doctor, and then doctor treats the patient. Because in my idea, it was like 23andMe. You do a test, we give you a lab result, but it was never like that. So every time we had to talk, and then you provide them with nutritionist or doctor. So in like 70% of people, unless it's this biohacker who are healthy, you will again send them to a doctor. So that's why.
I can feel your frustration. Yeah, we have it also in our company. So time is up. Yes. Okay, so thank you very much
Thank
for that
you.
presentation.
Thanks.
And now we are going for the last presentation of our competition. So please, the floor is yours. Okay. Take them. Yeah.
Okay. Let's now talk about something most people don't, even though 900 million women are affected every year. That is vaginal infections. Yet, 42% of women are misdiagnosed by their gynecologists, and 66% of women misdiagnose themselves. For us women, incorrect diagnosis can lead to severe consequences that mostly stem from recurrent infections. It can be fertility problems, risk of miscarriage or preterm birth, and also not only medical problems, but also psychological problems like depression, decreased quality of life, and problems with intimacy. So that's why at Shealed, we're developing the first self-test that will be capable of detecting 2 of the most common vaginal infections, bacterial and yeast. The test will be simple. You will take your own sample, mix it with a little amount of liquid, put a few drops on the test cassette, and within a few minutes, there will be a clear color-based result that will tell you if you can solve this problem with OTC medicine, or you have to call your gynecologist to get a prescription for antibiotics. What is behind the test, the technology? So the bacteria and yeast that cause bacterial and yeast infections secrete these virulence factors that attack vaginal wall and trigger vaginal infections. We will detect these virulence factors so that we know and are sure that the infection is currently happening. How do we know it will work? Because bacterial vaginosis markers are already clinically proven and has been commercialized for point-of-care assays. And for candidiasis markers, these are studied as therapeutical and vaccine targets, so there is a strong diagnostic potential. We have already done an external patent search, so we confirmed preliminary freedom to operate, but we want to do full FTO assessment next. And after, we will fill for a patent application that will focus, again, preliminary, on one-step self-test, combination of detection of bacterial and candidal infections, and also a novel detection of ongoing candida infections using these secreted virulence factors. So we have already validated our laboratory proof of concept for bacterial assay, and right now we're finishing yeast assay. Early next year, we'll begin prototyping that will be done with in collaboration with in vitro diagnostic manufacturers to ensure that the test is scalable, cost-effective, and manufacturable mostly. Then with finished prototype, we have to enter analytical and clinical validation to get a CE mark. So the market entry is expected in 2029. We have competitors, of course, but they are not enough. Why? The most common ones are pH tests, which only detect, pH levels, and they are also influenced by multiple factors that are not related to infections. Antigen tests, on the other hand, detect only presence of microorganisms, and most of them are present in healthy vaginal microbiome. Other alternatives are expensive, slow, or require a visit to the doctors. Unlike our competitors, we detect only markers of active infections, and we cover both diseases, bacterial, yeast, and also mixed cases when you have both infections at once, while also staying affordable. With an estimated retail price of €20, the global market is over 18 billion euros. And within the first 5 years into market, we aim to capture 240 million euros of this market. We know that women are already comfortable with self-testing because of COVID, pregnancy tests, and ovulation tests. So we are standing on the intersection of two growing markets, which is femtech and self-testing market. Our go-to-market strategy, since we are still in a lab development, is still evolving. So we have 4 scenarios for go-to-market, but mostly we work with the first one, which is going to market ourselves with a strategic partner. And initially, we want to focus on Central Europe, where we will target our end users, which are women, through educational campaigns and pilots with gynecologists. And since the gynecologists also want to use our test in their offices, we will reach them through key opinion leaders. And we will also do workshops for pharmacists so they know how to handle these consultations in pharmacies. Next, we'll want to expand to Scandinavia, to Europe, and then market eventually US. We will operate a B2B2C model. We will sell wholesale to pharmaceutical distributors that will supply pharmacies, drugstores, telemedicine platforms, and also gynecological clinics. And as I said, users will be women with symptoms of vaginitis and also gynecologists. That is supported by an early traction among women, pharmaceutical distributors, and also gynecologists. We, the founding team, we are all scientists with experience in microbiology, molecular biology, and R&D in biotech startup. We also know that we have overlapping knowledge and skills. So we are supported by a growing network of advisors that help us with regulatory, finance, grants, with staying clinically accurate, and we have also some scientific experts. Although we want to hire someone who knows how to do business and finance in the future, and also to expand our key opinion leader network and hire some marketing experts for future market entry. So far, we've raised 250K in idea stage from a VC investor and in combination with grants. Recently, we won Technologická inkubace from Czech Invest in Czechia. And currently, we're raising 1 million euros to get us through 30 months of prototyping, analytical and clinical validation. And it will be also supported by grants, which we want to apply, mainly EIC Pre-Accelerator, which we will be applying for next week. So this is us. Thank you so much. And I'm looking forward to your questions.
Thank you very much for the nice presentations. So I ask everyone, so we'll go for the patent. What exactly should the patent cover in this case? Once more.
Go back because it was very fast. Yes. So this is obviously in the discussion because we need to do full FTO assessment before, but we want to, from the results we have now, we want to patent the combination of bacterial and candida infection in a self-test format. And also the most innovative part on our side is the detection of ongoing candida infection using these virulence factors.
Okay, and you will go on the level just it is candida, not on whether it is albicans or different candida. Just
no, it will be. It's the same with basically bacteria because we would like to. I mean, these virulence factors are produced by the basically the group, not the individual bacteria or yeasts. So we want to ensure that we will have a higher chance targeting other species as well, not to target these. But for Candida, we are not yet sure if we will be able to detect also other Candidas like glabrata or next. We have to check first, but these are minor.
Yeah, that's clear. Yeah. So do you think that you can develop everything for 1 million?
So the grant we have now is for prototype development. So the 1 million is for basically a co-investment. And also we want to supply this 1 million with grants. So it will not be 1 million, it will be a bit more, but we want to also do another round for the market entry, the seed round in 2027, 2028.
Okay, and have you discussed already with some distribution company that actually they are interested in it?
Yes, we've discussed since, as I said, we are a bit early for them, but we've talked to two local ones, one from Czechia, one from Slovakia, and basically they said that if the test would be available, they would sell it tomorrow because they believe there is a high need for that.
Okay, that sounds good. And that's all of my questions. And I also wish you good luck with finding people for the business and financial part. And now,
thank you.
Okay, not many questions I have. You said that second phase you go for Scandinavia. Why Scandinavia?
Yeah, because we believe that there is a higher demand. Basically, their healthcare system is more how to say. Basically, people are more prone to pay more for their health. So as we speak.
As to the manufacturing, is the manufacturers of the product in Slovakia European or
Yeah, so we have options, but we want to have this manufacture in Europe. We don't want to have manufacturers from another continent because.
Thank you.
Thank you.
So, Benjamin, are people in Scandinavia willing to pay more money?
In general, yes, I think that is true. That they are either willing to or able to, at least. So that might be true. But I'm wondering, what was your price tag? Was that $20 per, or 20 euros? Sorry, per test. Didn't you mention that?
Oh yeah, so I'm not sure if I heard correctly, but the retail price would be okay. Great, thank you. The retail price would be 20 to 25 euro, but the wholesale price for distributors will be 10 to 15, depending on.
And what would the production cost be? Have you calculated that yet? First, I understand now it's more expensive, of course, and then when you do scale up, it will be cheaper. But do you have any estimations?
Yeah, so it's a very rough estimate, but it should be around 5 euro in this small scales, but then it should go low, we hope.
Okay. How much would you rely on a patent for this? Because you mentioned that you intended to file a patent, but have not done so yet, if I understood you correctly. Does your business strategy, does that depend on a patent, not to be copied by any others, or where do you see that?
Can you please repeat that question? I'll move closer.
Whether you rely on the patent as a part of the business, or without the patent you don't have any business. Yeah, this was a question. No?
Mm-hmm. Yeah.
Yeah. Okay. That's a good question. If we rely, if there is a business without the patent, well, no, I think we will not be acquirable, basically, without the patent. But I think there is a high probability that we will get that. But also, we cannot rely on that because it takes a lot of time to get it. So. Yeah, we're trying to protect us with NDAs and stuff, but yeah.
But so my main question would be, because you mentioned in the beginning that you already achieved one award somewhere. I can't remember where. Has any of this been public yet? Did you has there been an abstract or anything from your your strategy that has been publicly available?
Can you translate, please?
Yeah, basically whether you publish anything either in scientific journal or publish anything from a strategy that is publicly available, whether someone can steal it, I guess.
Oh, okay, we did not yet. And yeah. So.
Good.
Okay.
Thank you so much.
Thank you very much. So I would like to thank you for all of the presentations. Now we have to think about that and decide who will get the award. And that's about it. So now we have a break for... Ako dlho máme break? For 15-20 minutes. And at about 12 o'clock we will tell who will be the winner. Not enough? So half an hour? What do you think? Even more? 45. So okay. So 12:30 we will tell who will be the winner.
So after some deliberation, we made the decision, and this is a strategic decision for us, for the committee. So actually, I come from the probiotic world. I used to work in the biggest probiotic producer in the world, a big corporation. That's where I come from, and I do for Martin, Microbiome, angel investment I did, and coaching and mentoring a new business, a startup. So that's where I come from. Actually, it was our idea to start a kind of a dragon den in the microbiome space in Slovakia. What we see, the microbiome area is an emerging opportunity for investors all around the world. So a lot of money is going into the microbiome field, and If you look at the map, actually nothing happens in Eastern Europe. I used to come to Slovakia in the last 30 years, have good friends, and we noticed that bright heads, good people here, good science. And I asked myself, okay, if there's so good people, why is there nothing happening in the microbiome space, business-wise, right? There are a lot of academic knowledge, plenty of academic knowledge, bright heads. But still, what does it need to bring people closer to the money people, investors who are ready to invest in some academic research? Nowadays, even EU funds projects that have some chance of being commercialized, right? But in the last 20 years, nothing much happened in this area. So we thought, okay, let's make a Dragon's Den and show people what investors, what angels like me expect actually from a startup. Somebody comes, presents an idea. What do we need to invest? And actually, I thought, okay, I come to Žilina, nothing will happen. Something back 30 years in time, but I was wrong. The presentation was very good, top level. We used to organize the biggest probiotic conference in the world where leading experts come. And I can tell you, everybody was on top level. And this was a surprise, a very positive surprise. That means that the ideas, what you have, can be presented. You can present it to an international investor community. That means that's very good. The information presentation is excellent. The question is the idea good? The problem is, the issue is, there are plenty of ideas in this world. Ideas are not missing. Everybody has thousands of ideas. I have thousands of ideas. You have thousands of ideas. The question is, which idea is investable? Which idea are other people who don't understand your research field are ready to invest millions of euros? Now, that's the big thing. And the people who come from academia, they research something. Usually they don't have an idea about this. So the meaning of this event is to give you the impression what the other side expects. You ask for money, investor asks for information, market information, characters behind the product, potentials. That's what we ask for. You have to present that also. The academic stuff can be clear. We have all academic experts to evaluate that. But the business side is very difficult because none of the idea owner comes from big business, right? So that's just the introduction. Now the talk starts. So the idea was, the presentations are very, very good. But it was not a high school competition where we evaluate the presentation quality, right? It was the content. As to the content, we saw 3 presentations that were big chunk of business. They have the potential, the first 3. All of them need a lot of money to come to the next stage. All of them involved artificial intelligence in one or the other way, right? We have only one issue with artificial intelligence, and that is that everybody can use artificial intelligence.
It's a black box.
At the one day. So this is a tool that is open to everybody. So I'm not sure that it is a competitive driver, artificial intelligence, because you can sit next to your desk and use this kind, 25 different artificial intelligence kind of models, and people do it. At the end of the world, they will come to the same conclusion. Or maybe in their conclusion, your conclusion is already involved already. So they are maybe ahead of you. But that's my personal view. So we think for the first 3 presenters, they need big money to come to the next level. More evaluation, whether it's solid, the idea, whether it can stand competition within 3 or 5 years, where artificial intelligence also develops. So what we offer for the first 3, we will present their ideas to our investor group. That means in Eastern Europe, there are people who have money who are ready to invest. But they don't invest in life sciences big time, out of pharma, right? So that means there are investor people, big money bags. We say, okay, we want to be in the life science business, but we have no idea what is interesting. Peter, Martin, Benjamin helps us to identify the ideas. So we will present the first 3 to our investors, saying, this These are solid ideas, need further analysis. Money is not the big problem. The problem is whether it's really a good investment or not. We think that the 5,000 euro wouldn't make any difference for the first three. The first three are going for millions of euros, and that needs more thinking. However, the fourth one, where you can differentiate between bacterial and yeast infections of women, they have a point where 5,000 euro makes a difference. This is not for a flight ticket, but we will require a use of this. We would recommend or request that you use the 5,000 euro because you want the 5,000 euro. You use it for a patent attorney to evaluate your patentability of your stuff. Because if you cannot patent it, then you cannot make a business out of it in the long term. So we will help you to find one who can do this job, or you can propose anybody. And I think 5,000 is quite around enough to ask an international firm to evaluate your patent. So for the first three, we acknowledge the big guys, big money request. We will present it to the big guys with the big money bags. And for you, the fourth, we like the idea very much. It was a simple, no AI involved, classical distribution and business model, which is easy to understand. There's a demand for it. It's an important new area too. And I think it can make a difference if you use it for a patent attorney. So that's what I wanted to say. And I hope that this event will be replicated next year too, because this can give everybody in this area an impression what's going on in the world and bring in experts from global experts. They will come if they feel, oh, we can help this area, these researchers, Slovakia, to come further in this area, right? So Innovia is doing a job, and we are happy to be here and add some spice to the Slovak innovation and development projects. And that's what I wanted to say. If you have any question, come to me. Maybe Martin want to say something too.
Yeah, it is very hard to follow up after you know. So once more, so we would like to give you a prize. So let's say come here.
Thank you.
Okay. Say, yeah, we have to make photo. You have to thank you to the world or anything. And
Okay, I'm great at public speaking, so thank you so much. No, I really don't know what to say. I'm really grateful, and my team will be really grateful for this opportunity because, as you said, patent application is something that we really need to address, and also the expert consultations will be handy for us. So thank you very much once again, and thank you for this opportunity.
come here, we need to make photos. Nice photos for the nutrient ingredients and so. So thank you very much for your attention and congratulations. And I am really glad that you all came, and I hope, we will have it also for the next year. We will promote through whole Europe, and we will see who will come, because this is very exciting field. So thank you very much, everyone, once again. And now we can go for the main program, because I will be speaking there, so you can go listen to me there.